Welcome to AP Biology. This video is on plant structure and nutrition. We begin by looking at the structure of a leaf. A leaf is made up of three different tissue types. We have epidermal tissue, ground tissue, and vascular tissue. The epidermal tissue would be the skin of the leaf. We see it on the upper surface and the lower surface. The epidermis of the leaf is going to contain openings called stomata, and these stomata allow gases to enter or leave the leaf. We know that carbon dioxide is going to enter the leaf and be used for photosynthesis, and oxygen is going to leave the leaf because it is a product of photosynthesis. Surrounding the stomata are going to be cells called guard cells that you see right here and right here, and these guard cells control whether or not the stomata are opened or closed. Most stomata are located on the bottom surface of a leaf where it's shadier because it prevents excessive transpiration from taking place. The tissue in the center of the leaf is the ground tissue. The ground tissue is referred to as the mesophyll, and the mesophyll is made up of two types of cells. One called palisade parenchyma and a second called spongy parenchyma. And these mesophyll cells are the cells that are going to be performing photosynthesis. If you look at them, they are chock full of chloroplasts. In particular, the palisade parenchyma cells will be performing more photosynthesis. Also within the leaf, we see vascular tissue. Here you see a vein running through a leaf, and that vein is going to contain both xylem and phloem. Xylem is used for the transport of water within a plant. Phloem is used for the transport of sugars within a plant. Plants contain many symbiotic relationships with other organisms in order to obtain nutritional benefits. One symbiotic relationship is symbiotic nitrogen fixation. We know that plants obtain nitrogen from the soil in the form of nitrates. These nitrates are created by nitrogen-fixing bacteria that live in the soil. Some plants naturally have these bacteria growing on their roots. The name of the bacteria is rhizobium, and the name of the plant that has the rhizobium growing on its roots are legumes. These are plants like peanuts. They're going to have high protein concentrations because they have a symbiotic relationship in which they have nitrogen-fixing bacteria growing on their roots, which allows them to take in more nitrates, more nitrogen than other plants. A second symbiotic relationship is mycorrhiza. We've learned about mycorrhiza in the past. It's a symbiotic relationship between fungi and the roots of plants, and we know that this fungi increases the surface area of the roots, which is going to allow the plant to take in more water, to take in more nitrates and other nutrients from the soil. We also see many parasitic plants. Parasitic plants supplement their diet, their nutrition, from their hosts. They don't have to perform as much photosynthesis. Here we see some common parasitic plants. Here's mistletoe. This is called Indian pipe. You can see how it's white. It doesn't need to photosynthesize because it obtains all of its nutrition from its host. Here you see dodder plant. Dodder plant is a type of vine. All vines are parasitic. They extract nutrition from their hosts. This is not considered a symbiotic relationship, but we also see many carnivorous plants. An example of a carnivorous plant is the Venus flytrap, and they consume animals, or insects in this case, to supplement their nitrogen needs. Plants obtain nitrogen in the form of nitrates and ammonium from the soil. In order to obtain nitrates and ammonium, plants rely on bacteria that live in the soil. Here we see the two types of bacteria. One is nitrogen-fixing bacteria. They take in atmospheric nitrogen and convert that atmospheric nitrogen into ammonium. There's then a second group of bacteria in the soil called nitrifying bacteria that convert the ammonium into nitrates. Plants can then take in the nitrates through their roots. Also within the soil is another group of bacteria called ammonifying bacteria. They are decomposers. They consume dead organic material, what we call humus, and they use that humus to create ammonium as well. Plants have the ability to take in both nitrates and ammonium directly through their roots. So you see that plants really rely on the different types of bacteria that are present within the soil to take the nitrogen in organisms and atmospheric nitrogen and convert them into ammonium and nitrates, the two forms of nitrogen that plants are able to take in through their roots. If you look at the plant root in this picture here, all these little projections are called root hairs. And root hairs increase the surface area of the roots, which will allow them to take in more of these nutrients. Once nutrients are taken in by the plant or are created by the plant, they travel on three different levels. Transport takes place on the cellular level, there's short distance transport, and there's long distance transport. Cellular level transport involves taking things out of the soil and bringing them into the root cells. One of the most important substances taken in by the plant through the roots is going to be water, and water is going to move through osmosis, movement from high concentration to low concentration. More specifically, in plants, water is going to move through water potential. The difference between water potential and osmosis is that water potential incorporates pressure. We know that plants have thick, rigid cell walls, and therefore they cannot lyse if too much water enters them. Water potential is going to be used to measure the direction of water flow in plant cells. Water potential equals the pressure potential, which is always going to be positive, and solute potential, right? The highest the solute potential can be is 0, and the more solute there is, the more negative this number becomes. So to figure out the water potential, we take the pressure potential plus the solute potential. Water is going to move from an area of high water potential to an area of lower water potential. The pressure that builds up inside of a plant cell is referred to as turgor pressure. The more turgor pressure, the more pressure is inside of a plant cell, the higher the water potential. So water is naturally going to move within a plant from areas of higher pressure to areas of lower pressure. In order to facilitate the movement of water into the plant cells have pores in the cell membrane and the cell wall called aquaporins. And these aquaporins allow water to be easily taken in by the plant. Once water has been taken in by the roots of a plant, it is then going to travel up the plant through vessels called xylem. A couple forces are going to be in play here. One is going to be turgor pressure. As water enters the roots, turgor pressure is going to build up in the xylem contained within those roots. As pressure builds up, that water is going to be forced up the xylem of the plant. However, turgor pressure can only push water so far because gravity is also pulling down on the water. In order to get water all the way up the xylem of a plant, to get water from the roots all the way up to the leaves that need the water for photosynthesis, the process of transpiration is going to be used. Right, we call this transpirational pull. Transpirational pull is made possible through hydrogen bonding. We know that water molecules are stuck to each other due to hydrogen bonds, and this leads to cohesion and adhesion. Cohesion is water sticking to water. Adhesion is water sticking to other hydrophilic substances. The walls of the xylem are hydrophilic, and therefore the water sticks to it. So within the xylem, the water molecules are stuck to each other. They're also stuck to the walls of the xylem itself. Water is going to evaporate or transpire out of the top of the plant. Since the water is evaporating or transpiring, and because all the water molecules are stuck to each other through cohesion and stuck to the walls of the xylem through adhesion, as the water transpires or evaporates, the water in the xylem is going to be sucked upward. This allows the water to defy gravity and travel up the xylem of a plant to the leaves that need the water for the process of photosynthesis. We call this transpirational pull, and this is the major method by which water travels long distances through a plant. So overall, water is going to move up the xylem using two forces. One, turgor pressure that is building up inside of the roots, and two, transpirational pull that is taking place in the xylem as water transpires out of the top of the plant. This transpirational pull is all due to the hydrogen bonding that takes place between water molecules. Plants need to very tightly control the rate of transpiration. Transpiration is both good and bad for plants. It's good because transpirational pull is used to get water up the xylem of a plant to the leaves that need it for photosynthesis. However, excessive amounts of transpiration would be very bad because it's going to dehydrate the plant. The plant can't use water for photosynthesis if all that water is evaporating out of the stomata of the plant. In order to control the amount of transpiration, the plant is going to use the guard cells that are on either side of the stomata. The stomata are the holes in the plant leaves by which gases can enter or leave, but they are also the holes in which water is going to transpire out of. These guard cells have the ability to control whether or not the stomata are open or closed. Stomata are going to open on cool, wet days. Stomata are going to close on hot, dry, or windy days. Right? Those are the three factors that are really going to increase the amount of transpiration that takes place. Heat, dryness, and wind increase the amount of transpiration. Guard cells open or close the stomata by actively pumping potassium out of their cells or into the cells. Remember that potassium is a solute. Wherever potassium goes, water is going to follow it. On a cool, wet day, potassium is going to be pumped into the guard cells. As potassium is pumped into the guard cells, water is going to follow the potassium, and these guard cells are going to swell up and become turgid. When the guard cells swell up and become turgid, the stomata open. On hot, dry, or windy days, potassium is going to be lost from the guard cells. And as potassium leaves the guard cells, water is going to follow it. Water is going to leave the guard cells. The guard cells are going to become flaccid. They're going to shrivel, and this is going to close the stomata. So you can see here that plants have tight control over the amount of transpiration happening. They do this by opening and closing their stomata using those guard cells that surround them. The stomata are going to open on cool, wet days, and they're going to close on hot, dry, or windy days. In addition to opening and closing the stomata, we know that plants mostly have stomata on the bottom of their leaves, where it is shadier, which is also going to prevent excessive amounts of transpiration from taking place. Water is going to move through the plant using the xylem. Water moves from the roots to the shoots. Sugars are made during the process of photosynthesis, and they're going to travel in the opposite directions. They're going to travel from source to sink. In other words, they're going to travel from the leaves where photosynthesis is taking place to the roots and other parts of the plant where that sugar is stored. Sugar is going to travel throughout the plant using the phloem, and the sugars inside the phloem are referred to as the phloem sap. First, how do we get sugars made in the mesophyll of a leaf into the phloem that's found in the veins in the leaf? Plants are going to use the process of co-transport. If you look at this picture here on the bottom right, here we see the inside of the phloem, and here you see outside the phloem where these sugars are going to be made. Using active transport, hydrogens are actively pumped out of the phloem using ATP. Right, this is a hydrogen pump or a proton pump. This builds up a hydrogen gradient outside of the phloem. Those hydrogens, or those protons, want to rush back into the phloem. In the membrane of the phloem, we see a co-transporter. Now, if you remember, co-transporters will only allow hydrogens to rush through if it's accompanied by another molecule. In this case, if it's accompanied by sucrose. Sucrose is the form of sugar that gets transported throughout the plant. So the proton pump is going to build up this hydrogen gradient, and as the hydrogen gradient rushes back down into the phloem, it's going to pull sucrose into the phloem along with it. So it's using the movement of hydrogens from high concentration to low concentration in order to load sucrose into the phloem, in order to move all that sucrose into the phloem. This is important because hydrogens going down the gradient is actually being used to load sucrose against its gradient. Once sucrose has been loaded into the phloem, it is going to move from the leaves down to the places of the plant where it's going to be stored. Phloem sap moves through a process called bulk flow or pressure flow. We know that the xylem and the phloem run parallel to each other, which you see in this picture right here. We know that water is traveling up the xylem and the sugar is going to travel down the phloem. Well, how do we get it to go down the phloem? Places where sugars are loaded into the phloem are going to have a high concentration of solute. Since they have lots of solute, that means they have a very low water potential. The water is going to diffuse from the xylem into the phloem in areas where the sugar is highest in concentration. This is going to build up turgor pressure inside the phloem, and this pressure that builds up pushes the sugars down the phloem. So water moving from high concentration to low concentration is going to build up pressure in the phloem, and that pressure is going to force the sugars down the phloem. We call this pressure flow. And that water is then able to diffuse back into the xylem where it can continue its journey. Some plants live in very dry, arid conditions, and this doesn't make sense because they would have a very high rate of transpiration. In order to live in dry, arid conditions, these plants need to have adaptations that allow them to survive and allow them to limit the amount of transpiration taking place. These plants that live in arid conditions, like cacti and other plants, are called xerophytes. Here you see some of the adaptations they have. Number one, they have small, thick leaves. In the case of the cactus, the leaf is actually found in the form of the needle. And they're going to limit the amount of transpiration. Since the leaves are not flat, they do not absorb excessive amounts of sunlight, and that's going to limit the amount of transpiration that takes place. Xerophytes also have a thick cuticle. The cuticle is a waxy substance that covers the leaf, and it prevents transpiration. The thicker the cuticle, the more water is held into the plant. These xerophytes have all their stomata concentrated on the lower leaf surface where it's shadier, and some of these plants even have the ability to store water. Like we know that a cactus stores some water. This is going to conclude our video on plant structure and plant nutrition.